Okay. Hey guys. So this is unit 3. We're talking about cells, what their structure is, why they're here, what they do, all the things. Um, so when we talk about cells, we talk about, sorry about that, the foundations of life. Okay. They are going to be the building blocks of all living organisms. And in prokaryotes, the single-celled organisms, the cell is everything. It's got everything in it. There are no organelles. It's got a nucleoid region which has the DNA strand in it, and a lot of that single-celled organisms are bacteria and things that are looking to attack other hosts and change the DNA of healthy cells to the DNA that they have, so that those healthy cells will continue to replicate, or the healthy cells that become infected continue to replicate and go from there. Um, so when we talk about cells, cells are um, organized in a hierarchy. So it goes from small to large. So we have cells are the basic unit. Okay, that's the basic unit of life, and cells have the things that living organism needs. Then, especially in our living organisms and animals and humans and things of that nature, we go with this hierarchy. And then the next step would be tissues. Tissues are composed of interconnected cells that all have a common function. Okay, they have this common function, means they're differentiated. The cells are differentiated and have very specific functions that they need to perform. You can't take heart cells and put it in with liver cells and expect the heart cells to do what the liver cells are expected to do. They don't have that function. They weren't made that way. Okay, so you have the common function of cells that come together to form tissues, and ultimately, it's the tissue coming together that forms organs: liver, kidney, lungs, heart, brain, skin, all of those things. What's up, guys? I don't know if I have any. Check. You guys are always allowed to come in. You don't have to run away. I'm doing a video, but that's fine. Fine. See you, kids. All right. So you have the tissues combining to form the organ. Organs, again, have very specific functions. Those organs come together to form an organ system: cardiovascular, endocrine, respiration, skeletal, all of those things. Okay. So those organs come together to make up an organ system. And it's those organ systems that come together and work together to form a living organism, especially among the humans and among the animals, that they all work together. So can you see cells? No, you cannot really see them with your naked eye. You can see a bunch of them together, like when skin flakes off and things of that nature. But microscopes make cells easier to see. I'm going to be honest with you. I'm just going to go over this quickly. You need to be able to know that light compound microscopes bend the light around it so it illuminates it, but it doesn't give it in the same detail that electron microscopes do. But we're going to go through these anyway. So microscopes make small cells easier to see. And then we talk about magnification and resolving power. These are the two parameters that are the most important when you're dealing with microscopes and microscopes. Okay, so you can see here magnification. You have this butterfly here. It's at 1:2. The ratio is 1:2. 1:1, you get a little bit closer. You can see a little bit better detail here. You can see that there are lines here and a little bit more clearly delineated here than you can see here. But then you go to 5:1, and now you can see literally the little hairs off the head of the butterfly. This piece right here, you can see the structure of all of these pieces here. And its magnification is the process of enlarging an object in appearance. Okay, you've done this through the ages of time when you've done any kind of biology cell type of um, labs where you have to like look at the cells themselves. So let's talk about resolution. Resolution, not only in microscopes, but also TV. Okay. So you have a radiolarian right here. You're a regular light is going through it. It illuminates it. You can see it, and you can see micrometers, but you can't really see the definition of it. And you can't really tell that it's three-dimensional. Here, with the electron microscope, 50 micrometer, you can see that this A is a three-dimensional sphere. You can see the detail on the outside. You can see the points on the outside of the radiolarian. And it's that resolving power, the resolution, that is able to give you the structure and the depth that you need to see to be able to see that this is a three-dimensional figure and that this is detailed and very detailed oriented. The better the clarity, the detailed image, the better you're going to be able to make out. So compound light microscopes bend the light around it. So this is what you're used to seeing, right? Transparent objects like cells must be treated with dye or a chemical stain to be able to distinguish the different parts. And you can see how this dye has come in. You can see all the little organelles here. You see the nucleus. You see the cell membrane that goes around. If this is a plant cell, that would also be the cell wall. And again, you see how all these things are, but you can't really see a lot, right? The light microscope doesn't really give you a lot. Whereas an electron microscope achieves higher magnification and resolution using beams of electrons. The transmission of electron microscopes can show fine detail within the cells. These are probably red blood cells because of the concave nature that you can see on the outside of the cell. And you also are able to see the three-dimensional piece of the cell itself. Okay, so scanning electron microscopes provide that 3D exterior view of the cell itself. All right, so that's enough about microscopes, resolution, all that stuff. So let's talk about cell theory. And cell theory is the underlying principle of biology. All cells are the basic units of life. Okay, everything comes from a cell. All living organisms are made from cells. All cells come from pre-existing cells. This is the part of the cell theory that people look at with maybe a little bit of skepticism. Because if this is a true statement, that all cells come from pre-existing cells, then that means we are all related and we've all come from the primordial ooze, okay, the slime, so that we've all come from that. We've all come from single-celled prokaryotes, all right? So keep that in mind. Keep that in mind that is a big part of the cell theory, all right? So cells have four common components. One, they have an enclosing plasma membrane right around here, around the outside, around the outside, around the outside, around the outside. Okay, it keeps the outside out, the inside in. The cytoplasm is this gelatin-like substance that's inside the cell that suspends the organelles like the ribosomes and the nucleus and the centrioles and the endoplasmic reticulum, both smooth and rough. Okay, the cytoplasm is made of cell, which is like a semicolon in which the other components of the cell are found. Those are the organelles. Okay, in the nucleus, the DNA is in the nucleus in the big cells. In the single cells, it's in what's called the nucleoid region, because in there. And then fourth thing that all cells have as a common is the ribosomes, which you know what the ribosomes have. What do the ribosomes do? They make proteins. They synthesize proteins. Transfer RNA brings the amino acids to the ribosomes. Our RNA helps make sure that the ribosomes put the amino acids in the right order to form the polypeptide chain, which ultimately will become folded, and it'll either be a chain or it becomes a beta-pleated sheet. And the protein then goes through the different stages, primary, secondary, tertiary. All proteins go through the first three stages, and after the tertiary stage is when they become viable, and only the most complex of all proteins go through the fourth stage. All right, so the characteristics of prokaryotes. Prokaryotes lack that membrane-enclosed internal compartments. They don't have any of the oh my God, I forgot the word. They don't have a nucleus. They have what's called a nucleoid area where the DNA strand is located. They don't have any organelles. Organelles was the word. All right. So prokaryotes lack that membrane enclosed. Oh my God, I forgot the word again. Um, organelles, which are those internal compartments. Nucleus, the as we are all those things. Prokaryotes do have a cell wall that contains peptidoglycan, okay, which means it's a stiff cell wall that is then surrounded by a capsule. You'll see it on the next slide with the picture of a prokaryote. And to have that cell wall helps protect the bacteria even more, because as soon as bacteria goes into a living organism, it's going to be under attack because it's considered to be an invader in this. Okay. So prokaryotes are believed to be much like the very first cells that were found on Earth. Most of the prokaryotes are in the domains of archaea or bacteria, and most of them are those single cell. Okay. It's one of those, they also live in probably the most inhospitable areas of like water volcanoes, most archaea and bacteria. So when you're looking at a generalized structure of a prokaryotic cell, so here it is. You have your chromosomal DNA, which is inside, and all of these things. And it's localized in a nucleoid area. There isn't a, remember, there aren't the organelles there. So because the organelles aren't there, there's no nucleus. So it's called a nucleoid region in a prokaryotic cell. There are ribosomes that are in the cytoplasm, again, because protein needs to be made. You can see the cell membrane, as you have right here, the cell membrane here is surrounded by the cell wall, and then the cell wall is surrounded by a heavier capsule that's on the outside. Okay. So the other structures shown may be present in some of the prokaryotes, but not most of them. And usually bacteria will have some sort of flagellum or even this pili. The pili actually allow the cell to grab on to then able to inject itself into the cell, becomes a not healthy cell, and then once that's in place, that cell can then replicate. It has a direction in DNA from the nucleoid to continue to replicate itself, and that's how we ultimately get sick. Healthy cells, and that's how ultimately our system gets overwhelmed, and then we get sick. Okay. All right. So prokaryotic cells are smaller than the regular eukaryotic cells. The surface area to volume ratio is more favorable for moving material in and out of the prokaryotic cells, specifically the DNA, and they lack modifications found in eukaryotes that aid in their. All right. So here we're talking about surface area to volume ratio. So here you go. Surface area at one side. What do we know about cells? So in terms of this, it's got six sides, right? Because it's one on the top, 2, 3, 4, 5. And the volume is 1. Okay. You have two sides here. Okay. Two sides. The surface is going to be 2, 4, 6, 8, 10, 12, 24. The volume is 24. The surface volume is 24 divided by 8. And then in C, the sides, you can see 3 cubes here. 3 times 3 is, or 3 squared times 6 is 4. And then the volume would be 27, 3 cubes. And again, your surface area to volume ratio would be 2 to 1. So you can see the ratio, but the volume. All right. So you can see it here. You have 1 by 1, very small volume. But you go to 2 by 2, and you see how the volume increases itself. All right, we're going to stop there for now. What's up? Do you guys need me? Want to need me? Yeah. I thought I saw Lori say yeah. Give, I only gave you the one. that was given to me. Yeah. Is Lori okay today? What's going on? Like she is hell on wheels today. Good. That's what makes you did. Now you're going to be hurt. You're injured. You're out.